So we're going? I have a super tiny thing I just wanted to follow up. Jeff has the main topic today. But I kind of overshot a point that I was making when I was talking about how, like, yeah, we give up some accuracy and stuff when we sparsify quantize and so on. But like, I look forward to being able to then, regain that. I wish I hadn't drawn this dot higher than that. What I want to be able to do is achieve the same thing with something cheaper. I'm not going for anything better. Just reclaim this.
So you weren't claiming that we might do better. You just
Right.
basically want to get back to where we were before,
Yes.
but with less resources.
That's the point I wanted to make, and I literally overshot it. I shouldn't have gone
If
further.
we had there, you did it in a minute. That's amazing.
Yeah.
That's
Yeah,
great.
that's my entire topic.
Although, if accuracy includes robustness,
That's
then
another
that could
axis,
be a whole
right?
different story.
Here's Florian.
Yeah, I mean, yes. I guess I mean accuracy in the big sense of how well the networks do. And
Yes, I would include robustness in that bigger sense.
For sure. I mean, I just called this cost. That means all sorts of things.
Oh.
No, but in that bigger sense, I would put the dot back where it was.
Oh, okay, I see. I
That's
see
what I
what you're
mean.
saying. Okay.
Well, or you could differentiate as a third axis. Accuracy or robustness. Or
Yeah, but at the same, there's a trade-off between how good this is and how expensive this is.
Right.
I feel like you can always collapse down to those two axes in some conceptual way.
All right.
And
okay. So some figure of merit. Okay, right.
that's it. That's all I had.
All right. Florian just walked in, so we can get going. I'm going to go over the stuff I talked to you about yesterday, Florian. It shouldn't be very long. So I'm going to do it. So yesterday I started reading this paper, The Cosine Directional Tuning of Theta Cell Burst Frequencies, which I don't remember if you reviewed the whole paper or just talked about one of the figures in it. I can't
I didn't review the whole paper, but I mentioned the two key figures in it.
Okay,
to make a point.
so I read this paper and another paper that I'm going to talk about here, and I had an insight a couple of insights which I thought were really interesting, and I was confused about something. I talked to Marcus first, and then I discussed this with Florian, so Florian knows most of what I'm going to talk about. So the basic idea I'm going to present is quite simple, and it relates to the cortical column model. I'm going to end up talking about that again at the end, but there's a lot of background to it, so I'm just going to run through a few things. First of all, this paper's primary purpose was to show that there are it was well known that there are theta cells or theta bursting cells in the hippocampal complex in different places, and but the theory of the oscillatory interference requires that those cells have different frequencies depending on which direction you're going. They have to be directionally tuned. So if you're moving in one direction, the speed up in the theta frequency will be faster. If you're moving slightly off, it's a little slower. If you're moving orthogonal, it doesn't change. These people, this is a 2009 paper, I think, or 2011. They basically said that hadn't been shown before. So the main purpose of this paper was to illustrate in animals that there are cells that are directionally tuned, and that as the animal's running, the increase in the theta frequency is tuned to the direction of the animal. That they claimed it hadn't been shown before, and that's what this paper was about. So I'm not going to review all that. So they basically said, here, we're showing it. And I thought this little figure here is useful to see. This is a little recording from a single cell. And what we're talking about here is cell goes through these little bursts, and the bursts occur at the theta frequency. So that's what we're measuring when they're like, this is like 8 hertz or something like that. And then they showed basically in different parts of the brain, old brain, they showed that what these figures are about, they're showing that they actually are directionally tuned. So we're going to skip over that. Yes, we found directional tuning cells. There's some interesting stuff there, but I want to get to this figure, which Florian brought up and was talking about, and I was really confused when Florian was talking about it. Nothing to do with you, Florian. I just wasn't. There's a lot going on here that can easily trick you up, and especially this lower part here with these little rings and these rings up here. I didn't understand what the rings were, and I have a hypothesis. That's the point of today. I'm going to relate what's going on in this picture to what I think might be going on in the cortex. Let me just walk you through this. This first part up here, A, is a little confusing too. They're showing a rat moving across this one-dimensional track, and this is all theoretical now. There's nothing. This is all model. There's nothing biological here. And they say, what if we had 8 different theta cells that were directionally tuned. As the rat is running at 25 centimeters per second, then some of these will be a little bit faster than other ones. So the theta 8 is going to be the fastest frequency, and theta 1 is the slowest frequency here. You can barely see that. This position along here has nothing to do with the position along the track. It's just that the animal is running at 25 centimeters per second. So that was fooled me too. I thought somehow this had to do with where the animal is. No, it doesn't. So at this rate, you would see these cells oscillating as frequency, different, slightly modified theta frequencies. And then they point out if you just look at any 2 of these cells, just 2, you will see an envelope waveform like this, which is grid cell-like. So, because they come in phase and out of phase, in phase and out of phase. And so as the animal would be running along the track, you'd see activity, no activity, activity, no activity, activity, no activity, and so on. So that's what that was about. Then
That's summing up the 2?
It's just summing the 2. So if you sum the 2, you get this black oscillation, and they're in phase, and then they're out of phase, and then they're in phase, and then they're out of phase, because they're slightly different frequencies. So it's like this wah, 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 as you have a background frequency. And so the idea is that during the top here, that's when you get those little bursts of spikes. You get the little bursts of spikes here, and you get the little bursts of spikes here. Actually, that's not the early burst of spikes. Sorry, this is like a grid cell, a one-dimensional grid cell. So if you just took 2 of these oscillations at different frequencies as the animal was going along, you would see, and you took a cell that looked at those 2 and summed them up. That would be the what that cell would look like. This thing then got me really confused. And I had to ask Marcus clued me in what was going on, because same frequency, it'd be like all of them are theta 1 or theta 2 or something like that. And yet they're slightly out of phase. So they're all oscillating at the same time, at the same frequency, but they're doing it slightly out of phase. The example I give is it's like a wave in a stadium, right? So everyone's standing up and sitting down in the stadium at the same frequency, but the actual peak of a wave is going around and around. So this is going to actually go around here within 1 theta cycle. So an 1/8 or 1/10 of a second, all these cells are going to go woo, 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 and they're all like going up and down, up and down, up and down, right? Made up. Okay. So that's gone continuously. It never stops. Unlike a head direction cell, we'd say, oh, it stops moving when the animal stops. This never stops. It just goes around. These cells are going like that. Then they said, well, if we had a whole bunch of these cells, so now, and then they came up with this chart, and this really got me confused. It took me a while to figure out what the hell's going on here. This lower picture here. So let's just walk through it. These columns represent cells that are directional orientation. So everything above one of these guys is a directional orientation, the same directional orientation, meaning they will speed up or slow down proportional to how much they're moving in that direction. Okay? So that was what he was talking about earlier. So if I just look at one row of these, that means I have a bunch of cells that are cycling through different peaks at the same frequency. But these would be the frequency at which they're going around would be different based on which direction you're heading. Right? So if you're heading in the preferred direction, that would be the fastest cycling around, because the theta frequency would be going faster. And if you're in the antithetical or the anti-preferred direction, it would be the slowest one going around, but it's still going around. This direction here is the sort of baseline speed, the vertical axis. It's like, how quickly does the speed increase based on how fast the animal is moving? And what I think, and I confirmed with Florian yesterday, this is not a continuous function. These are basically like the step functions you see in the entorhinal cortex, where you have a grid cell module that's at one scale, and then another grid cell module at another scale, another grid cell module at another scale. So this is not a continuous function. So this might be 1.4 times faster than this, and it's like a different grid cell module. Okay, and I've talked about in the past, I don't think it's correct to look across these, and I've made the argument that this is equivalent to like V1 in cortex and V2 in cortex and V4 in cortex. That is, they're sort of separate they're working at different scales. And so within the hippocampal complex, there's going to be mapping of rooms that work best at different scales. But they're not necessarily interacting with each other. Okay. Now I'm going to get to the denouement of the whole thing. I don't know if it's any questions about this yet?
Is it possible that the modulation would be less than 1? Because it looks like there's a slope that's less than 1.
They talk about this in this paper, so it's a good question. I hope everyone heard it. The idea is you have a base frequency, and if you're moving in the preferred direction, it goes up. But if you assume that in the non-preferred direction it would go down, then if you look across all these cells, the average rate would stay the same, right? Because you got some are slowing down, and so if you look at the local field potential of the whole thing, you wouldn't see everything speeding up. You'd see something slowing down, something speeding up. And they point out that that's not what observed in an animal. What's observed in an animal, as the animal moves, everything seems to get faster. So I'll show you one more picture, as I said I was going to show you. This one up here, yeah. This is a tricky little thing they're doing here. What they're saying here is, and there's evidence for this. This is both speculative, theoretical, and observed. That if you look at any particular cell, there's a sort of a base theta frequency everyone's got. And as the animal starts moving, all the cells get faster. That is, the base frequency goes up. And relative to that base frequency, some get even more and some get less. So that's what's going on in this upper right drawing here.
Different slope.
Yeah, so they're like, the circles are, here's like the animal's not moving, this is your base frequency. Now the animal starts moving, the base theta frequency goes up for everybody. That's these circles. But the ones that are up here, see it says preferred and anti, the preferred one goes up even more and the anti ones go down a little bit. There's a little subtlety there.
And that's cosine tuned, as you see in the figure C. So you get a 0.2 hertz increase if you're heading in the right direction, and like a minus 0.2 hertz decrease when you're going in the wrong direction. So it's a very small change.
Yeah.
But that's actual experimental data.
Yeah.
So this
and we need a small change, because the way, if you want these, imagine the animals running along a track, and we're talking maybe 50 centimeters or 100 centimeters between maybe some certain grid cell, the cell firing once and firing twice. It takes a long time for that animal to get that 50 or 100 centimeters. And what we need to do is these cells have to get in and out of phase at a slow, they have to be so close in frequency that it takes that long for them to get in and out of phase again. You know what I'm saying? So the differences between these frequencies have to be very small. And that's what's observed. By the way, this figure here was problematic for me, this B. I wasn't going to mention it. They're showing variation in how these cells perform, and the variation is quite significant. So I didn't understand this.
Yeah.
They're saying here, like you take, this is one cell, this red dot here, and they're saying this one cell sometimes could represent something as small as, let's say, a 30 centimeter spacing, and another time it might represent like a 75 millimeter spacing. Like, it didn't really explain that. So I don't know if I understood that correctly or not, but it was a weird variation. All right, I'm going to come back now. More questions? That was a good question. Okay, just by the way, the reason they showed this picture before I would give my point, they're saying, look, if you just sample a couple of these, like here, you see these are grid cells here, right? They say and this guy says, if we just sample from, a few of these rings that are 120 degrees apart, so this ring and this ring and this ring, you'll end up with a grid cell. If you have a cell that looks at those, that cell will have this sort of grid cell firing. And up here, we're assuming that they're moving faster, so they'd be closer together, that kind of thing. They also make the point that if you sample across these, which I don't think actually is going to happen in the brain, you get border cells. And they make this argument, if you look at all these cells, you get place cells. I don't think that's right either. So I think really the thing that I think is right here is these grid cells. And they're pointing out that you can get grid cells by sampling from these ring attacks. I know there's several levels of interaction going on here. That's why it's so hard to keep it in your head. At least I found a way.
So maybe I'm thinking of this wrong, but I have a question. So it seems to me that the base rate of how quickly the wave's going around the stadium doesn't really play a role in the scale. doesn't play a role in how close, for example.
No, it's how quickly it increases when you move.
Right. It's like what really matters is how the phases of these different things change.
Well, here's the way about it. Imagine I have two stadiums, right?
Yeah.
And they both have the at the
Yeah.
same time. They're doing the same speed.
Yeah.
And now the animal starts moving. Well, the wave goes around faster, but in one stadium it goes around faster than in the other stadium. They both get faster, but one gets faster still.
Yeah.
That's the difference. And
The thing that matters is how much faster the second stadium is than the first. But it doesn't matter if the base rate of the two doesn't matter. It's just the
in fact, I don't think that the two stadiums aren't even being compared here. This is another weird thing. I don't understand this yet. What they're saying here, they're not getting into this issue here. I'm confused about this. I need to read this further. But you remember the oscillatory inference model was about having a base theta and an increased theta, and that was to show the precession or the of the peaks. They're not even talking about that here. They're saying here there is no base theta. They're not even talking about a base theta. They're not comparing these two.
That is actually the difference between the very earliest 1D model that I showed, which was this idea that there's a base theta and then a higher one on the dendrite input. Whereas the later models, like for example the hybrid model that I showed, doesn't have a base theta, so to speak.
Yeah.
But it uses pairs of oscillators in opposing direction. And if you can combine those, then you don't need a base theta.
Okay, I need to understand that. I don't yet. I understood the words you said, but I need to get the turn. But in here too, they're not really talking about that. They're just saying if you hypothetically propose that there are these cells that are all going at the same rate and they're all going in perfect synchrony, like the wave in the stadium, which was made up. Right? They just made that up. Then you could sample from them, and if you sampled from like 3 of them that are really like 120 degrees apart, and you said this cell here looks at this blue cell here, which is the same as these blues. This blue cell, all these are the same cell. This is where they're the same cell where it's responding in an area. If that cell looked at this cell and this cell and this cell, it would have this pattern of firing in the environment. Okay, that's what they're saying there. It doesn't explain the precession at all. It just says I would get a grid cell if I did that.
Excuse me. So when you're saying that there would be an opposition that would be 180 degrees phase apart, I don't see any connections.
No, this is, well, here you go into 2D, as Florian was talking about recently, the optimal was 120 degrees.
That's why the best hybrid oscillatory interference model has 6 inputs. Because you get...
Oh, okay. So
there's
Yeah.
different ways to get to the all the different things you need, like the grid and the theta precession. And the simplest possible model is just assume a somatic theta and then a faster input. But that's not the only way to get there. And so it's...
Okay.
Yeah.
okay. Thank you for the.
All right, now we're going to get to the point of today's talk, today's discussion. I asked myself, I said, well, there's some compelling things about this. And some of the things they thought were compelling, I didn't find compelling, like how you could get a place cell out of this. I don't think that's right at all. But I said, this is an interesting way of generating grid cells, and it seems there's some data behind it and so on. I said, but where are these circular cells? Where is this ring attractor? Have we seen anything like that? Where would that exist? This requires a whole bunch of cells that are sort of working together. They're all working at the same frequency, and yet somehow they're taking their turn, like the wave in the stadium. If that were true, it's got to exist someplace. And I'm thinking, like, where would it exist in the neocortex? My first guess, which I think is wrong, was I said, well, I'm always looking for a roll of a mini-column. I said, well, what if we're just talking about one section of a like a one layer. In fact, these are just all in one layer of the hippocampus. It's not everywhere. So I said, well, what if it was like in here I had these cells like this, and they could be firing in progression, firing in progression. I don't think that's right. Then it occurred to me that there's another possibility, And the other possibility, and I'm not sure if it was the order, if I read this paper first or if I thought of it and then read the paper, I can't remember now. The other possibility is the following. Let's go back to the classic ice cube model in the neocortex. And the classic ice cube model in the neocortex is you've got, if I go across in one direction here, you see these orientation changes. And as I've argued recently, I think we have misinterpreted these, that these are not spatial features. These are, almost all these cells are directionally sensitive, and I think these are, in essence, these cells act like a dot product between the movement and your preferred. They represent a preferred direction of movement of the eye or the finger. This sort of organization exists everywhere in the cortex. So these are essentially, you can use sensory input to derive how something is moving. And I can go into it further if you want, but I think these are like the little things in the bottom of that figure down there. The directionally oriented, and as you put a sinusoidal grading in front of the animal's eye or across their finger or something like that, what you'll get is varying response rates of these things depending on how if this is mapping directly to the movement or if it's angled and then nothing, right? So. I think that is the right way to interpret mini-columns, and I'm going to have to develop that into a further theory. However, there's always been a mystery. For those of you who've been in this room for a long time, know that I've always wondered if, when you look at this block, that when you move in one direction, this is of course like a millimeter cube of neocortex, top layer, bottom layer, we're looking to the side here. Down from the top, you'll see that as you move in this direction, you go across these orientations. But when you go in this direction, it's all iso-orientation. It's the same orientation. I sort of show a little mini-column here, but if you go look at the mini-columns in this direction, if you go in this direction, all of them have the same orientation. And I've always wondered, I said, why would you do that? What's the point of having the replication here? Why have all these things the same going in this direction? It always bothered me. And there's never been any explanation or a proposed explanation I've ever heard of. So you might recall, I've wondered, I've proposed various weird things that never worked out for that. Then it occurred to me, well, I wonder if what you're seeing across here is there's this traveling wave across here, which is the changing in the phase. It's like this is the stadium unwrapped, right? This is this little circle of dots, but it's unwrapped here. This paper, called hippocampal theta oscillations or traveling waves. This was 2009 by Lubinov and Siapis. And what they show here, and I won't go through all the details of it, there's a lot of methodological stuff that they did to get this to work. But what they basically show in the hippocampus, not here, but in the hippocampus, if I were to look at a cube of the hippocampus, I can do that better. Look at a cube of the hippocampus, that what you see is the theta oscillations that are occurring in waves that travel in one direction. And so the wave is going this way. The wave in the stadium is traveling across the surface. And if you look carefully, and this is they've well documented now. I'm not going to go through all the details. I've interpreted this figure, and I'm not going to try to interpret it for you right now. But what it basically says in my world is that you get a complete 360 degrees in about 1 millimeter. Okay, this is in hippocampus. This dimension here in the cortex is about 1 millimeter. And so it occurred to me that if the same mechanism that was happening in the hippocampus, where these theta waves are moving across, that could be happening in the cortex. And what's going on in this dimension is you have a series of mini-columns that are occurring at different points in that circular stadium of the wave in the stadium. So these guys the cells in this slice all operating at the same frequency. All these mini columns in these slices are all operating at the same frequency. That frequency is tuned to the movement of of this sensor, whether it's part of the eye, the finger, whatever. So it speeds up and slows down from the theta rhythm, but it's unraveling. It's this traveling wave across here, and so now that provides a potential explanation for why you have this slab like this, and it gives you all the information that they're proposing you need in which to build a grid cell module system. I've never heard of anything like this being observed. And of course, when the early days when Hubel and Wiesel and all those people did this work, I don't think they were thinking about these cycles at all. They just didn't do that. So if this were true, this is a strong prediction. It's measurable. You can do it in, they've shown that you can do this with these multi-tetrode arrays. That's what this paper does. They use a two-dimensional multi-tetrode array that they can plant on top of the cortex or the top of the hippocampus, and they can detect these traveling waves. You could do the same for the neocortex, and I'm giving this a fairly high likelihood. I wouldn't say 99%, but I'd say greater than 80% chance that you would see that traveling wave there. And now, what's exciting to me now is it gives me, for the first time, I can start building out perhaps a more complete picture about, it's just a big clue as to what the hell's going on in a cortical column. If we assume that this architecture, which apparently is seen everywhere, I have to go back and look at Mountcastle's papers, because I think Mountcastle actually said that this slab idea exists in all modalities. That this might be how the cortical columns are structured. One dimension is the change in the orientation of the movement. That's what it is, the orientation of the movement, and the other is this traveling wave. And now you have the structure for building everything. So that was the thought for today.
Could I ask on that left-hand ice cube picture, what would be the direction where you cross across ocular dominance?
I'd have to go back and review that, the ocular dominance issue. I don't remember, Kevin. I'm obviously familiar with it, and I can't remember, but I don't remember the details. There's also, can I bring up a related topic? I'm going to admit my ignorance. I don't know the answer to the question. But there's something interesting to think about as well. What I'm proposing is that most of what we used to think like, oh, grid cells are driven by motor behavior, and they are. That is, the animal moves in the dark, the grid cells get updated. But I also have shown that grid cells get updated, they must get updated by pure sensory input. Like the example is, again, if you're sitting watching some video on the screen and you're like a video game, you're in someone playing a video game, and as they move, as the person playing the game moves around, your sense of where you are in the game changes too. You know that I moved forward, I moved over here. And so there's obviously the grid cells representing where you think where this player is in the world out there in this game are being updated purely by sensory information because I'm not doing anything. And so that was one of my conclusions. And I said to myself, I think that's the primary way that the system knows movement. And I argued that now it's the same thing as happening with your finger. When you move your finger over an object, there's sensors that detect the movement in the direction and how fast you're moving. And that's how, and that the mapping to motor is later learned. Okay, so now let's put it this way. So if I think about a finger is really just I can only move, the surface of my finger can only detect movement in 2 dimensions. But that's not necessarily true of the eye. The eye could learn in 3 dimensions. And because all it requires is if I have this field that's expanding this way, I know I'm going into the field, and if my field is going this way, I know I'm going out of the field, but if the field moves like this, I'm going left and right. So it occurs to me that in vision, unlike touch, where we have this, that we could take advantage of the not only the 2 eyes, but also the flow in and out. And so if that were true, then you would expect to see in visual cortex, if I had cells that are not just sensitive to movement left and movement right, but into and out of the field of view, And I think there are cells like that
I
there.
think they are in either like MT or MST or something.
Okay, so
Those,
I'd be surprised if they weren't in V1 as well.
Could be.
What do you mean into out of field of view?
Like these cells respond, they get active when you have something moving at a certain,
okay.
right, moving across the field.
Okay.
What I would expect some cells that say, no, I only get active when things are moving like this,
Yeah.
or moving like this.
Yeah.
You know, in Star Wars when they go into light
Warp
speed
speed.
and it warp
Yeah.
speed. And it
Yeah,
goes-
that's right.
I
So
don't know.
like the stars gives you the idea of going forward, right?
Yeah. So
That's part of the link to flow.
this is very interesting because we really want to map three-dimensional structures, right? And it's tricky in touch because it doesn't appear that I can sense in 3D movement in 3D. So I have to have some additional mechanisms. And Marcus has talked about some things we might do, but in vision it might be that I really can sense movement in 3D directly in the sensory system, and therefore I would not want to just have my orientation columns, which are really my movement columns. I wouldn't want them to be as 2D. I would want them to be 3D. I want them to be representing movement in this direction and that direction. You know, things moving around as I move like this and so on. So I would expect to see in cortex columns, and you said they're there, so that's good. I would expect to see columns that are directionally sensitive in 3D. They're pretty the same idea, but they're moving in and out of the plane. And therefore, we need to learn n-dimensional maps, and so this will allow us to build an n-dimensional map. We could, instead of just having two-dimensional orientations, we could have three-dimensional orientations.
You're talking about mini columns that are selective in three-dimensionality.
No, each column is sensitive to movement in 1D, right? They have a 1D preference. So this would be like a 1D movement preference. And what we'd want to do is you'd have other columns that are sensitive movements in outside of the plane.
Oh, right.
Remember we talked about, like in the past, I've talked about how these columns the spatial pooler would automatically, if it would automatically find vectors that would cover the entire space. And it could be two-dimensional, it could be three-dimensional, it could be n-dimensional.
Right.
We want a general purpose mechanism that says, you just give me some inputs, and I will figure out how to map the n dimensions of that input as best I can into a grid cell-like representation, and then I can start building n-dimensional models of whatever it is that I'm looking at or sensing. So that's the goal. And these guys, even in the end of this, in the end of the cosine directional tuning paper, the very last thing they talk about, which is interesting, I think it was in this paper. Was it this paper or was it the other one? Oh, here we go. Let me see if I can find it. One second, bear with me. They basically say, this is I'm going to paraphrase because it's complicated. They say this could be a very general purpose system to map any kind of time varying state variable x, which essentially could be any n-dimensional thing. Here we go. It basically says, in principle, the existence of such oscillators in the nervous system would make it possible to synthesize almost any desired envelope function in the state space of X, thereby provide a powerful and flexible mechanism for constructing mental representations in biological neural networks. Basically, X represents sort of an unlimited number of properties and dimensions, and they're saying this could be, I'm reading between the lines, I'm saying that they're suggesting this is a very powerful mechanism that could be used for lots of different things. That's what they are saying. It could be lots of different things. I'm not sure if they're thinking the same things I'm thinking, but they say this is a general purpose system. So we really would love to come up with like a really general purpose methodology for how a column in cortex can learn the structure of anything. Anyway, this idea that this theta thing, the stating wave moves along one dimensions of the cortex, I found fascinating. I did realize since yesterday, after I talked to you, there's a little bit of weirdness to it, and I'll point out the thing that I think is weird, that it gives me pause. in the theta wave paper, they show this like just a line of activity that's moving all simultaneously across. Like there's this traveling wave going across the surface of the hippocampus. But here, if we're talking about these guys are going to have slightly different frequencies, right? Because at any point in time, some are going a little bit faster, some are going a little bit slower. And therefore, there isn't a single wave going across here. The wave that's going across here is going to be at one frequency, and the one that's going across here is going to be that frequency plus some delta, and a small delta. And so there's going to be sort of there's going to be a slight, there's going to be a gradient of frequency as you go across this way, because there's a gradient of frequency that you're going across this way. I haven't rocked that yet. So what does that mean? Does that mean the wave sort of the wave sort of like bends a bit? Does the wave not go straight across and it starts looking like like this as it crosses? I don't know. It's an interesting thought to think about. I haven't really internalized it yet, but it's not a killer. It's just like an interesting thing. It's like, it wouldn't be just perfectly straight lines going across. It would be kind of warped, right?
If anything, that's almost
I'm gonna go to the meeting.
Okay.
that almost supports the theory because it makes it where these waves would be harder to find unless you were looking for them.
Right.
Something like that would explain why neuroscientists haven't discovered this already.
I'm not sure if any neuroscientists actually. I don't.
Maybe they have.
Maybe they have. I'm not even sure anyone's even looked for it yet.
That's right. But people would have accidentally found it after 50 years of neuroscience.
Well, okay. Well, my point is, the 2009 paper said no one's ever looked for traveling waves. We're going to look for traveling waves. So.
One other thing I'll just throw in because it's useful to throw this out at some point. Someone has really been banging the drum of traveling waves in the past couple of years. Terry Sinyovsky has some recent stuff where he has a big review paper from 2018, I think, where he's just reviewing traveling waves in cortex or traveling waves in the brain.
I highly recommend. He just gave a talk at the Simmons Institute. It's actually Sherman is in the audience, and it's like an interesting conversation.
This was the brain amputation thing.
Oh, I spoke at that conference.
Okay, great. Okay, cool.
I don't remember.
You weren't at that talk.
I don't remember. I couldn't go all of them. There's
But he talks about traveling waves. I want to review the underlying paper of that because it speaks to this.
Can you send me that paper?
Yeah.
Or want to do?
Yeah.
One of the nice things now is I have a lot more time because on Monday I finished the book. So I still have things to do, but so right now, it was wonderful because just yesterday I came in, I started reading papers instead of writing. I hadn't done that in a year.
You were sitting in your other chair.
I know, because I was like,
new life.
oh my God, I don't have to write this morning, And already I felt like great. Look, I made some progress in the first few hours. It was like wonderful.
Can I mention just two things that I'm thinking about?
Yeah.
I'm not going to take this far.
So, excuse me, will you send me that paper?
Yes. Yeah, you send the paper, I send the video.
Send us the research, I want to see it too.
Right, okay. So two things that I'm thinking about is one is that this model, because there's this this directional organization, right, in your cortical ice cube. The question is, right, for one, is it really true that the typical orientation of traveling theta waves in your cortex, which do exist and which Terry Sinyovsky talks a lot about, actually is sort of oriented the right way to work with this?
That's good. That's part of. I first of all didn't know that it was known to exist, so now we know because there's a whole study already. But yes, the prediction here would be you'd see it in this.
Exactly. So it needs to be perpendicular to the orientation changing of the mini columns.
It doesn't say anything particular about what should happen when you go in this direction. That's not all I should know when going in this direction is the frequency should change. But I can't say anything else beyond that.
And only during movement should the frequency.
Yeah, during movement. I'm sorry. During movement, the frequency would change, but I'm not sure that. I'm not sure. I don't.
I think that's misleading. I think it's misleading to start the discussion about cortical grid cells with the autometric primitives of the navigational system in entorhinal cortex and hippocampus.
Why is it a mistake?
I think it's a mistake.
Why?
Because, because if you're thinking of the Constantinescu paper, for example,
Yeah.
right? So you want to get away a little bit from like navigation, right? What you're really after is not necessarily, movement speed in some direction. I understand that is important, whether that's a moving finger or a moving eyeball or, or your moving legs.
Yeah.
But in order to be as generic as possible, what you want is simply a column that detects, right, preferential change in a, encodable dimension. And if that happens
Yeah,
to be
totally,
like
of
legs
course.
and necks,
Of
then,
course.
right?
That goes without saying. But the nice thing about thinking about it here is there's a lot of data on this cortex. Right? There's a huge amount of data. We can go look and read about what goes on in somatosensory cortex exactly like this. And so I think obviously we want a generic mechanism. And so, but I think also the reason I'm focusing on the sensory stuff is because I think this is a really big idea that I think everyone misinterpreted what orientation columns are. I think it's a complete misinterpretation that we think of it, and I was fooled by this too, that it's actually a spatial feature. It's not. It is basically dividing up movement through a grid cell space or through some space, and that would be true whether it was early vision or Constantinescu birds. It doesn't really make a difference. But I think by including primary sensory cortex in it, which no one is thinking about today, nobody's thinking, as I know, is thinking that stuff may even occur in V1 and V2.
Right.
That's why the Constantinescu paper, they looked at,
Ventromedial
prefrontal cortex someplace.
Yeah.
Like, people don't think V1 does anything. People think V1's just like some filter. So,
That's true. They treat it like a convolutional network.
yeah. So I think bringing that into the picture, because there's so much more data here. Like this kind of architecture is known. I don't think anyone knows what the prefrontal cortex looks like in terms of this kind of structure.
Right.
But yeah, you're right. The point which we need to genericize this to say, well, what is the Uber principles going on that if I stick this on top of an eyeball, I get this, but if I stick it on top of some other cortical outputs, I get birds.
Just to check it, and with your mental model, like here you're drawing the ice cube version of this, like the Hugel and Wiesel version.
Yeah.
But often the pictures in the cortex are more like pinwheels.
Yeah.
Do you basically just adapt this and picture it like spiraled around into a pinwheel?
At the moment, yes. Without any, there may be more to it. Right? Do the pinwheels really represent some other type of computational function that I'm missing and I don't know about yet? I don't know. And they probably do, but I don't know what it is yet. So I'm just going to go where I can go. So there are parts of the cortex that really do look like this. And there's other parts where they're kind of spiraling into those vortex points, right?
Right.
The pinwheels. So I don't know what the hell's going on there.
I guess I don't actually know, but I was thinking that, in fact, all of it is pinwheels in actual cortex, actual V1 of primates. And this was just like the Hugel and Wiesel initial guess.
No, I think I could be wrong. I believe if you actually look at the cortex in the dimensions that it is, that you can easily pick out 1 square millimeter things that look just like this. And that the pinwheels are sort of...
We should be certain about that.
I've seen a lot of the papers where they've got really lots of bands and crazy things.
Yeah. We could go look. It's just a question of dimensionality.
You can select it and get that.
Well, the question is 1 millimeter small enough that you can isolate sections that are basically orthogonal like this versus incorporated?
I think that's true.
I think it is too, but I'm not 100% certain of it. But it's really easy to check. You just got to find those pinwheel pictures and look at the dimensions on them.
The nice thing about the pinwheel picture is that you don't have a critical point where it's a starting point and traveling out to some boundary. If you're circulating around, you don't have that. The reason why I brought up the mention of the ocular dominance is that if we're really looking at this stuff instead of features as a set of oscillators and their differences, well, the difference between the ocular dominance gives you 3D directly.
Yeah, the reason I tend to dismiss ocular dominance is I ask myself, well, what can I do with one eye? And there are very few things you can't do with one eye. So I could watch the video game with one eye and have a sense that I'm moving in and out of the screen and all that stuff. So that doesn't require 2 eyes. Obviously, 2 eyes adds advantage. It could give better depth perception, for no doubt about it.
But in particular, one of the effects that we use to create the illusion of depth is to vibrate things just a little bit, where things in the foreground vibrate more and the things in the background vibrate less.
Yeah.
This type of structure can pick up that kind of phase difference as a spatial organization, not requiring, wholesale movement, but just the fact that I'm getting, this amount of amplitude of oscillation here versus this amount of amplitude of oscillation here.
But that's the kind of artificial thing too, I think, right? Well, I'm not sure I even need that.
But it is one of the things to create the illusion, in 2D of a 3D.
So remember, there's two things going on. One is, where are you? Not just it's 3D. It's like, where are you? What the thing you're looking at? Where is it? Where is its location? That's not the same as 3D. I need to have a three-dimensional representation of space, but I'm trying to locate where something is. And I'm moving, and I'm trying to do path integration to say, well, if I know where I am now, and all of a sudden I move like this, and now I know I'm in a different location, that's not just the same as saying I'm perceiving a depth. That is somehow, you're inferring movement through space through the sensory input. And generating the concept of 3D is not generating movement through space. It's just saying, oh, that thing's further and this thing's closer.
Right, you can do it from a static position.
Yeah, so we're trying to get to the point where we're saying it's like grid cells. It's like the rat moving in the space. We're trying to imagine us. Well, we're trying to imagine us moving through space, and moving. Whether I'm doing this or this or this. I'm trying to update my grid cells based on sensory input. And I don't know yet if I know I don't need the two eyes to do that.
I'm trying to take one of the key points that you just put out, is that we've been thinking of these as feature detectors, and what do we do when we do computer vision? We find features and then try to compute their disparity. But if what we're seeing here is really these oscillations, and just by the fact that from microseconds or whatever, you get this little disparity kind of going on, you could actually pick that up and bypass the whole notion of collecting a bunch of features and computing their disparity, and saying, are there phase differences between these co-mingling sets of I'm looking at the same place type of thing. So that's
You're talking about figure ground separation? I'm confused about
Something like that, yeah.
Yeah.
I'm saying you
That-
can
Sure, I get it.
judge distance with this too.
Right,
Yeah.
right, and I'm saying that the oscillatory model of the phase differences gives the capacity for eliciting that signal locally,
Yeah.
and you have to globally put it together, but you're getting that differential
Right. You
signal.
I think-
learn it.
I think-
You can learn it.
This is only. I'm gonna end this because I'm running out of steam here. But this is only part of what we need here, right? This is just one small part of what we need. This is just basically trying to create a reference frame for where the viewer is in the world, and changing that reference frame where the viewer is in the world. That's all that this is accomplishing at this point in time. There's all these other things. There still has to be sensory input which gets assigned to these locations. That's still occurring here. And there's other issues about how far away something is. This doesn't calculate that. This just tells me where my eyeball is. So there's a lot more going on. I just feel like that this is a big clue. That's all. And it's not the answer to everything. It's just like a piece that could be really important. And so now that I'm going to try to build up a much better model of what's going on, at least in terms of this sort of grid cell stuff, and that will become a substrate in which to figure out further things. It's just a step. But it's an exciting step for me because it's the first time I've ever had an idea what might be going on in this dimension here.
Isn't there also, I think we looked at fetal development, particularly I think in cats, that there's these traveling waves that move across.
Yeah, across the retina.
Right.
Yeah.
And that's an interesting training.
Yeah, that's what it's. I think most people think that's what it is. It's,
Yeah. So I think
Setting
traveling
up
waves
the
are.
topographic organization of V1.
Right.
It's well known,
yeah,
I think.
Yeah. All right. Unless someone else wants to go into this further, I just wanted this. It's supposed to be a really short topic, but I knew there was a lot of background to get to it. But that was it.
Okay. Thanks everybody for watching our research meeting. Bye.